Welcome to Let's Chat About, which is a free webinar monthly series that Sophia Sees Hope is running in 2021 as yet another initiative to connect with and help educate our inherited retinal disease and Leber congenital amaurosis communities. My name is Alyssa Bass. I am the Director of Marketing and Communications for Sophia Sees Hope. And today we are welcoming our friend Ben Shaberman from the Foundation Fighting Blindness. Ben is the Senior Director of Scientific Outreach and Community Engagement for the Foundation. He's been with the Foundation for about 16 years, and he is an expert in helping people in our community understand inherited retinal disease and Leber congenital amaurosis. The Foundation is a wonderful partner to Sophia Sees Hope. We do lots of programming together, and we donate to research through the Foundation. So we are grateful as always for the Foundation's support. We did take questions through the registration process. So we have a number of questions that were submitted in advance. We are going to use the Q&A function within the webinar for people to submit questions. You can do that during our conversation. We will take questions at the conclusion. We have turned off the chat function, so you can ask questions just through the Q&A. This is going to last between 30 and 45 minutes. One of our goals in doing this is that it's not a day-long event that requires a huge amount of time. So we're going to dive as deeply as we can into this very complicated series of topics, but we're obviously not going to be able to cover everything. We are recording this session, and it will be available afterwards, probably within a few days, and we will send you the link when it's available. So Ben, welcome, thank you. Before we jump in, I just want to take a couple of minutes to talk about you. You have a bachelor's degree in computer science, a master's in systems management, and a master's in poetry. And you've been with the Foundation for more than 16 years, where you lead their professional outreach team, educating eye care professionals and members of the communities about resources for people with inherited retinal disease. So thank you for joining us.
Thank you.
So talk about your road to what you do now, because I just think it's very interesting that you're a poet and a novelist and also can write scientific content.
Well, thanks, Alyssa. And first, I just want to thank everyone for joining us today to hear about the research. It's always a privilege and an enjoyable thing for me to communicate the research that's going on out there around the world to address these challenging conditions. It's fun to be the messenger, and I appreciate all the great work that Sophia Sees Hope does to bring families together and to drive the research. And thanks to all of you out there who support the Foundation Fighting Blindness as well. So, sort of a long story short, I got out of college wanting to be in technology, sales, and marketing. And so that's what I did. I sold mainframe network processors and did some systems engineering, and did that and related sort of data communication stuff for the first 10 to 12 years of my career. And it was interesting, but it just didn't move me. And I ended up getting a master's degree in poetry in the evenings, just as something to exercise my creative interests. And I had written poetry in high school. And anyway, so long story short, I leveraged that into medical writing and healthcare writing. I actually worked for a long-term care facility, a couple hospice organizations in the late 90s and early 2000s. And lo and behold, I was looking for a new job, and I literally stumbled across the Foundation Fighting Blindness. They had a science writing position. This was in fall of 2004. I knew nothing about the eye. I knew nothing about the retina. I am not affected by an inherited retinal disease. I don't have a loved one who is. And I just thought it looked interesting. And so I dove in, I got the job, and it's just one of those serendipitous things where, as Dean Bach said, a researcher from UCLA, who I heard early on in my career, you can sort of get seduced into the retina because the science is so compelling and fascinating. And I really was. And so here I am, 16 years later. And one little interesting anecdote about that journey, when I started, I was just writing about mouse studies. That's basically all I did. And not being a scientist, I was like, well, that's great. We're curing blindness in mice. Is this really going to move to people? And so in 2006, 2007, when that started happening, that was a huge moment for me personally, like, hey, this stuff really works. And now we have 40 clinical trials underway for emerging therapies. So it's pretty cool.
That is pretty cool. So we have attendees here today from industry and from research, but we have the vast majority of people who are attending this webinar today either have LCA or care for someone with LCA. So just give us an overview of what is LCA and how do the conditions affect the retina?
Sure. So I think the best way to think of LCA is a family of inherited retinal conditions that cause significant vision loss in early childhood. That's the common denominator. And I think the family of disease concept is very important. LCA can be caused by, as we know up to this point, about mutations in about 27 different genes. So it's very genetically diverse. And it's also very clinically diverse in the way it affects the retina. So some forms of LCA, depending on the genetic mutation, may affect how a photoreceptor functions. It may affect how a photoreceptor develops. It may impact the vitamin A processing cycle, the biochemical process that makes the retina light sensitive. That's actually what Luxterna addresses, and we'll talk more about that. Some forms of LCA cause severe loss of retinal structure very early on. Other forms of LCA may cause severe vision loss, but some retinal structure remains. So not only is there diversity within each gene, each bucket, I guess you could say, genetic bucket, even people with the same mutated gene can have different experiences. So I think it's incumbent upon people to become knowledgeable about their or their loved one's specific situation and obviously advocate for that person.
Go ahead.
I just wanted to mention, there are about 8,000 people affected by LCA in the US. That's sort of the estimate, and obviously there are thousands more around the world.
And it makes it easy to understand why it's very hard for people after their initial diagnosis to figure out where to go and what to do because of all of those different genes and the different ways that they react and the different ways that you'll have symptoms and what you will go through. So what should families do when someone in their family is diagnosed with LCA?
Sure. So it's important to have a good doctor who hopefully you're in a community or able to get to a doctor who understands LCA to some extent. Your typical private practice retinal doctor may not. I guess some pediatric ophthalmologists and retinal specialists may or may not. But it's good to stay in touch with a doctor, get regular exams, even if there's nothing from a treatment standpoint, just to manage any complications that may be related to the LCA or something else. And I think, as most people know, genetic testing is usually warranted to get to understand what mutated gene is causing the disease. That really gets at the root problem and will help put you on the path to what treatments and cures that are emerging may apply. The foundation has a no-cost genetic testing program. It's on you can learn about it on our website. I know the good news is in this group, I think most of your constituents know that pretty well and pursued genetic testing. And the good news for LCA is that in a vast majority of cases, they can find the gene. So there's good success in genetic testing. In the case where the gene isn't identified, it's good to be in touch with a genetic counselor to talk about retesting and the potential path forward if you don't know the gene, because there is a path forward even if you haven't identified the gene early on.
And some of the forms of LCA have other health issues related to them. So that's another reason why it's important to, it's not, in some cases, it's not just vision.
That's correct. And I'm glad you said that. Some of the cases can be syndromic depending on the gene and the way it's mutated. And genetic testing, even if those other issues aren't apparent in a young person, genetic testing can give you some clues as to whether it may be a syndromic situation. So yet another good reason to get testing. Good point.
So you mentioned Luxterna in the beginning of our conversation. Give us a little overview of Luxterna. It's mice to dogs to people. So give us a little overview of the evolution of Luxterna, and then let's talk about how Luxterna has impacted the rest of gene therapy research.
Sure. So the Luxterna journey, I think you could say, started in the early 90s, both when Jean Bennett and Al McGuire had a vision that gene therapy could be a way to treat these conditions, even before the RPE65 gene was identified, before we really even had any genes for IRDs identified. So they were already thinking about gene therapy. The gene RPE65 was identified in 1993, just as a gene that had function in the retina. It was a couple of years later that they learned that mutations in RPE65 could lead to LCA. There was a mouse model created, some natural history studies, but the big moment came in the late 90s and in about 2000 when Briard dogs were identified as having RPE65 mutations, which basically caused the same form of LCA as in humans. And at Cornell, University of Pennsylvania, the researchers there tested a gene therapy for those dogs, and it had dramatic effects, vision improvements. And that was the first aha moment for what would become Luxterna. So that was the first 7 or 8 years. That ultimately led to a clinical trial being launched at Children's Hospital in Philadelphia. Again, Jean Bennett, Al McGuire, in late 2007. And a few months later, the first results for that trial were reported. Young adults had some significant vision improvements. And as that trial went along and they started treating younger patients, the improvements were even more dramatic. Kids putting away their canes, seeing the faces of loved ones, some seeing stars in the sky, really dramatic improvements. And that was the first real breakthrough in the field of inherited retinal diseases. So CHOP spun off Spark, the biotech, in 2013 to raise the money to commercialize the therapy, because those later phases of a clinical trial are very expensive. Spark did that. Luxterna was FDA approved in December of 2017 as the first FDA approved gene therapy for the eye or any inherited condition. And the treatment has continued to perform very well in people who are receiving it as a commercial product. Obviously, it's an important thing for RPE65 patients, but to the rest of us, involved, it's really provided affirmation to researchers and companies that gene therapy is the way to go for many of these conditions. Not all forms of LCA. I think that's important for people to understand. Gene therapy isn't the only answer, but it is a pretty cool and elegant way to address these conditions, replace the bad gene with a good gene, in essence.
So one question that we did receive in advance was, do you think that we will see changes to Luxterna, the existing successful treatment? Do we expect an evolution in that, or do we just expect new treatments for other genes?
So Luxterna as a product isn't likely to change. Once you get FDA approval for something, you put a stake in the ground, you're probably not going to change it. I don't want to speak for Spark, but that's my educated guess. But keep in mind that Luxterna is sort of old school gene therapy technology because it went into the clinic in 2007. So gene therapy technologies have continued to evolve, and pretty dramatically, to be safer, to work better, to penetrate cells better. So all the other gene therapy trials that have been launched since then, most are using newer technology. And I know at the foundation, we're funding newer technologies all the time. So you don't just stop with what you have, you keep making it better and better and better. But Luxterna itself will probably stay the same, and that's okay because it works pretty darn well.
So that's a great segue now to really the big question that we're here today to answer, which is, tell us about what's in the therapy pipeline. And as you're telling us, if you could explain the difference between gene therapy and gene replacement and CRISPR as we go along, I think that's helpful too, because sometimes these terms get confusing.
Sure. And the information I'm about to provide, I've got a laundry list here, so bear with me. But before I talk about these genetic therapies, mostly gene therapies, is to understand that gene therapy is not going to be the answer for everybody with LCA. And that's going to depend on the gene and how that gene has affected the retina. There are other emerging therapies like stem cells and optogenetics and neuroprotection, which operate independent of the mutated gene that may be more applicable to somebody with an advanced form of LCA. For example, optogenetics, there are a few clinical trials underway for that. That targets other cells in the retina for restoring vision, cells other than photoreceptors. And so for somebody who's lost all their photoreceptors, gene replacement therapy is not going to be the answer. But if you can harness other cells, that might be an effective way to restore vision. Likewise, Tom Ray, a stem cell expert at University of Washington, is working on a way for the retina to grow its own new photoreceptors, which is a pretty cool thing. They sprout from Mueller glia cells. Not ready for the clinic, but it's gene independent, something we're funding, and that may apply to somebody who can't benefit from gene therapy. So with that, I am going to start talking about some of the other gene and genetic related trials and preclinical efforts that are underway. And first, Procure, our partners in the Netherlands. So they have an approach called an antisense oligonucleotide. It addresses the mutation in RNA, which are the genetic messages. And they have a therapy in a phase 2/3 for a specific mutation in the gene CEP290. And this mutation is actually very common in the LCA world. It occurs in a specific intron. In the phase 1/2, they had really nice results. Many of the participants had improvements in visual acuity, mobility, and full field sensitivity. They've moved it into a phase 2/3, which is great. That is ongoing. And hopefully, sometime perhaps later this year or early next year, we'll hear about results for that. So that's an RNA therapy, not a gene replacement therapy. Another company is companies are targeting CEP290, that same mutation. Editas and Allergan have launched a clinical trial for a CRISPR-Cas9 gene editing therapy. You can think of that like molecular scissors to cut out the mutation. Again, CEP290, that's in an early stage clinical trial, phase 1/2. They have not reported results, but that's the first CRISPR-Cas9 therapy that's ever been administered to directly to the human body. So again, a very cutting edge thing, and it's happening in retina.
Now that's the one that got put on hold because of COVID. Am I right? They had just started their trial and then COVID hit, and you couldn't be scissoring people's genes in an operating room.
You know, I am not sure. I know a lot of trials were delayed because of COVID. I'm not sure if that was the case, but
okay.
Perhaps. And that's another important point. A lot of people out there, you may know a lot more about things that I don't know about, as Alyssa, you're inferring here. So I'll do the best I can to tell you. Now, another company who recently launched a gene therapy clinical trial is the startup Kcnj. And they were co-founded by Shannon Boy at University of Florida, longtime foundation-funded researcher, wonderful researcher. And she and her colleagues have launched a trial for people with GUCY2D mutations. That just got off the ground in 2020. She recently mentioned that they finished dosing the first cohort of patients. They haven't reported results yet. So stay tuned for that. You know, one of the nice things about GUCY2D, and this applies to some other forms of LCA, is that even though there can be severe vision loss in the patient, they can retain some retinal structure that's amenable to gene therapy. So there are cells left that you can try to save. I'll add that the Foundation Fighting Blindness is funding that through our venture philanthropy fund, the RD fund. MiraGTx, a company in the UK, has also done work on a gene therapy for AIPL1, which is a particularly severe form, or causes a severe form of LCA. And they are actually offering a gene therapy for AIPL1 for compassionate use. So the thing there is the gene therapy is probably only going to help children who are very young, and what I've heard, 5 years or younger. So if that happens to be your case, I would definitely reach out to MiraGTx. If you reach out to me or Alyssa, we can put you in touch with them. But that's a compassionate use situation. It isn't something that is an official commercial product, but you could get access to it. MiraGTx also has an RPE65 gene therapy in clinical trials. They are working to move it from phase 1/2 to a phase 2/3, and stay tuned for any news on their efforts to move it to another, actually the phase 3 trial. So that is an overview of the clinical stuff that's underway. Now I'm going to talk about several preclinical efforts that are underway. Another biotech, Iveric Bio, is working with University of Massachusetts on a CEP290 gene therapy. So this is gene replacement therapy. It's for the whole CEP290 gene, or at least all the mutations. It's not targeting one mutation. So replace the bad gene with the good gene. Because CEP290 is a big gene, they're developing what's called a mini gene. They're taking out parts of the gene, but still with the goal of keeping the gene fully functional so it can help people with all mutations, but they try to shrink it down so it fits in the viral containers that are used for gene delivery. The French company Harama earlier in 2020 announced a collaboration with Leiden University Medical Center on a CRB1 gene therapy. That too is not in a clinical trial. They've been talking about getting a clinical trial underway in perhaps 2023. CRB1 is something that the Foundation Fighting Blindness has focused a lot of research on. Jan Winholtz in the Netherlands has been somebody who's been behind this particular effort quite a bit. And there are other investigators like Jeremy Kay at Duke and the lab at University of Pennsylvania who are also focusing on this gene to understand it better and to potentially come up with other ways, potentially other gene therapies for CRB1. For sake of time, I'm not going to get into the nuances, but gene replacement can be challenging depending on the gene, because the gene can express itself in many different cell types, and there can be different ways it expresses in different people too, depending on the mutation. So there can be a lot of variation with this in the same gene, I guess is the message there. Another gene that we've gotten questions on, has come up across my desk, is the gene CRX. CRX is a little different from a lot of LCA genes because it causes dominant disease as opposed to recessive, which most LCA is inherited recessively. And CRX is actually linked to a lot of other retinal diseases as well, not just LCA. But there is a startup company in France called Variant that's working with a research group in Paris and in France to develop a gene therapy. They're still in a kind of an early stage as a company. They've shown some promising results in mice. So they still have some ways to go. Again, an early stage company, but that's good news there. There are other things that have gone on with CRX, some stem cell work at UCSF. There's actually a cat model of CRX disease, which can help with therapy development as well. Another gene that's getting some attention is Kcnj13. The researcher, I'm looking at my notes here, Bakash Potnick, and I apologize if I butchered his name, a researcher at University of Wisconsin-Madison, is working with a small startup company, Hubble Therapeutics, to develop a gene therapy for Kcnj13. And we're also funding Dr. Saha, also at University of Wisconsin, for a gene correction approach using nanoparticles. He's looking at that in both human cells and mouse models. He's still at a relatively early stage for that. Another gene that's gotten some attention is LCA5, laborcillin. The researchers at University of Pennsylvania recently published, this is May 2020, on some promising results in gene therapy in mice for LCA5, for laborcillin. So that's good news there. They've also done some nice work in characterizing patients. And they've also reported that even for some people, at least with LCA5, who have severe vision loss, there may be still an opportunity to restore some vision thanks to some remaining retinal structure. Also on the preclinical radar screen is MNAT1. We've gotten some questions about that. At our Visions conference in the summer, Eric Pierce reported some nice work that they did in a gene therapy in a mouse model for NAT1. And that work continues. I don't have any further updates since Eric reported that information, but I know that that work is advancing. For NPHP5, there's been some nice work at the UPenn Vet Lab in canine models. Again, that's a condition where there is some retinal structure that remains despite severe vision loss, and they've had some encouraging results in canine studies. RDH12 is a gene that's gotten a lot of attention over the years, especially at the University of Michigan. Deborah Thompson has done some nice work in mouse models. Again, some gene therapy work there, and there's been some encouraging results from that. They've also done some natural history studies to understand how mutations in RDH12 affect people. And in addition, Moorfield's Eye Hospital, Maria Musagi, has done some work in characterizing LCA caused by RDH12 mutations. And I know there's an interest in moving that work toward a clinical trial. And then finally, we got a question about the gene TULIP1. And earlier in 2020, there was a research paper that came out from Trinity College Dublin. They conducted a gene therapy study in mice for TULIP1 mutations. They noted that the expression wasn't quite what they had hoped for. They need to go back and try to improve that. But I think it's important for people to understand that a lot of these studies are iterative. You don't just treat a mouse, it works, and you're done. You often have to go back a few times to make sure you get it right and optimize it before you start thinking about moving it in humans. That's part of the reason research takes a long time, even in the lab. So those are the genes that I had on my radar screen, and I did a little research on, or perhaps I already knew about what was going on for those. And it's a lot of good work going on out there.
And it can be an overwhelming tsunami of very technical information for an average person whose pediatrician says to them, something's wrong with your baby's eyes. So how can families find good, scientific, reliable, truthful information about LCA in general, and then as they move through the process and they understand their specific diagnosis for their gene itself?
Sure. Well, the Foundation Fighting Blindness website, fightingblindness.org, which I develop content for, is a good resource. I can be a resource, or other people at the foundation. If you're bold and you are willing to look at research papers, which can be a little daunting if you don't have a science background, you can go to PubMed and do some searches there. And some of the papers aren't fully available. Some are open access. And if you know a good doctor, that can be helpful. And then obviously attending events like this and other events hosted by the foundation, where you can have interaction with the researchers and ask specific questions. Those are good ways too. So yeah, it's work. I mean, you have to be your own advocate or your child's own advocate, but more and more information is becoming available. So that's the good news.
Let's just talk for a minute before we open it up to questions, because the Foundation Fighting Blindness and Sophia Sees Hope funds research, including brand new research that is just getting started. Can you just briefly talk about how the foundation determines which research to support and how that works?
Sure. So we have a scientific advisory board of retinal experts from around the world that evaluate research projects that are proposed to us. And they and other leadership in the foundation are working to identify gaps in the research, where the need is. And through their collaboration and a grants submission process that we conduct every year, we try to identify and evaluate the grants that have the most scientific process and at this promise, and that are addressing the gaps and the needs. So need is important to us, and good science is important to us. And the good news is we're well connected. We know the space. We're really working hard to understand those gaps and those needs, and who the best researchers are in their projects to fill them. The RD fund is an important aspect of our research plan because we're working to get more treatments into clinical trials through that RD fund as well. We also have a translational program to get things closer to the clinical trial doorstep. So we're funding research at the whole continuum: lab research, research to get it close to a clinical trial, and then research to get it into a clinical trial. That final step is attracting investments from biotechs and big pharma who can cross the finish line by investing tens and hundreds of millions of dollars. That's our strategy. And right now, it's working relatively well.
Yeah, based on everything that's moving through the pipeline, it sounds great. So thank you to Carol DeFranco for answering the question that Editas did pause its clinical trial because of COVID, and they have restarted. So they are again working on patients in that clinical trial. So thank you, Carol, for that. We will open it up to questions. We do have a question in the Q&A. Let's see, are there any electronic devices, apps, or wearable devices one could use to remotely monitor the clinical trial endpoints in CEP290 without burdening the patient with an in-person hospital visit? Which endpoints could possibly be measured by a wearable in such LCA10 studies?
That's a great question. And I will confess it's a little out of my area of expertise, but it is a question that our clinical development group at the Foundation Fighting Blindness is exploring. And the idea would be that through a watch or an app on your phone, you could potentially track somebody's activities of daily living, or maybe even conduct some kind of retinal, basic retinal test or vision test remotely. And it would be really great to feed that data not only to a clinical trial, but perhaps a registry where that data could be accessed, not the personal information, but the endpoint information could be accessed by researchers to understand how a therapy is working. Something as simple as, is somebody spending more time outside at night after receiving a therapy because it's helping them see better at night? That's not such a hard thing to do, to understand through an app, but it's a valuable endpoint. That's just an example. And I'll say at this point, it's something we're interested in, and the technology is getting there. Now, in terms of an actual clinical trial to do that, I leave that to the regulators, because that's where you need the FDA to say, yes, we will allow that type of monitoring as part of a clinical trial, or no, we won't. And I confess, I don't know the FDA's view on that at this point. So I think you have Wiley Chambers coming up for your next talk.
We do.
Great question for Wiley.
Nice plug. Thank you, Ben.
Sure.
So that was the only question that was in the Q&A. Do you have any, we've gone 41 minutes. Do you want to give us some words of wisdom, some concluding thoughts, and then we'll say good afternoon?
Okay. I guess the only other thing I wanted to mention is our patient registry, My Retina Tracker. I'm happy to report that we have about 800 LCA patients and families in My Retina Tracker, in our patient registry at myretinatracker.org. And for those of you that don't know, My Retina Tracker is a patient-controlled registry. Patients and families put information about their retinal disease in the registry, And the de-identified data is available to researchers and companies who are doing lab research or natural history studies, or perhaps most excitingly, clinical trials. And they use that information to help guide their clinical trial development and design. And they also use it for clinical trial recruitment. So it's important to be in My Retina Tracker if you want to be on the radar screen of the research community. We have to remember these diseases are rare, and the more people we can get on that radar screen, the better massive information we're providing to the research community to inspire them to develop treatments and cures. And the other thing that's important is if you're in My Retina Tracker, it's important to provide as much information as you can to keep your record up to date, because the more data you have and the more current it is, the more valuable your record is. So in short, My Retina Tracker is a great way for you to basically raise your hand and say, I or my loved one has LCA, I'm on the radar screen. Please know about me so that you can develop therapies to help these conditions. And again, personal information is never shared directly with the researchers. You actually reach out to them if you match their search criteria. You're notified through us. They never get your personal information directly from
Right,
us.
it's safe. So give that web address again for My Retina Tracker.
myretinatracker.org.
Okay, and Foundation Fighting Blindness?
fightingblindness.org for everything else.
Okay, and
For
you guys
research news.
are on all the social media?
We are.
Okay.
We work hard to be out there and visible as much as possible.
And it's important to follow organizations like Foundation Fighting Blindness on social media because they are trustworthy and they provide real scientific information. Sometimes families can get caught up in, people try to scam people, and especially vulnerable people who are trying to make their way through a complicated diagnosis. So follow organizations like Foundation Fighting Blindness, like Sophia Sees Hope, for the real, accurate, true scientific information. Ben, you are amazing. Thank you so much for your time. And we will email everybody who attended the link to the video as soon as it's available.
Thank you, Alyssa. Thanks to Laura Manfrey and the rest of the Sophia Sees Hope team. You guys do a wonderful job. I always like working with you and being at your events when we can do that. You're a great family, and it's an honor to be sometimes a part of that family, as I was today. So thank you.
Well, thank you. All right. Have a great day, everyone.